Hello, dear students. We are discussing about this process of translocation through the tram trap and or even the what we call it as the translocon. The translocon is a combination of mm, two components. So here, uh, sorry, yeah, this one we are the one we are I was discussing about. This process is called as co-translational translocation. Why it is called as co-translational translocation? Because so here you can see the mRNA which is attached to the ribosome when it gets stalled within the cytoplasm during the process of translation. When it gets stalled within the cytoplasm, later the complex, the trimeric complex of ribosome, mRNA and the SRP comes and sits onto the SRP receptor. Later the release of SRP and its receptor takes place, leaving the ribosome on the translocon complex. So then the translocation is going to restart and it is going to continue till the translocation is going to be completed. So if you see the fourth step there, the SRP is leaving and the movement of polypeptide chain is taking place into the lumen of endoplasmic reticulum. So this process, as it continues, the translation is going to be happening. mRNA, the protein is going to be growing by decoding the mRNA molecule. On the other hand, you can see the nascent polypeptide chain which is being grown is transported or is being moved into the lumen of endoplasmic reticulum. So this translocation of nascent polypeptide chain which is being synthesized by the ribosome is called as co-translational translocation. So this co-translational translocation is an important step in the synthesis of secretory proteins which get secreted out of the cell through the secretory pathway. So then there is one more called as post-translational translocation. So if you remember, I was talking when I started to discuss about this particular chapter, I told you the proteins, in order to be functional, they need to get proper three-dimensional structure. So during which they undergo several kind of modifications. So in order to fold properly and to achieve a functional structure, the many times the proteins which get synthesized within the cytoplasm, there are a number of proteins which get synthesized in the cytoplasm, but still these proteins need to be transported into the lumen of endoplasmic reticulum, from there to the Golgi complex for further processing and as well as modifications. And this movement of a polypeptide chain which has got synthesized within the cytoplasm into the lumen of endoplasmic reticulum happens through post-translational translocation. That means in case of earlier step we see co-translational, where translational coupled with the translocation. Here this particular movement takes place only once the polypeptide chain synthesis completes within the cytoplasm. After that, that particular polypeptide chain, with the help of number of protein folding molecules called chaperonins, is going to be translocated into the cytoplasm of the into the lumen of endoplasmic reticulum. So this is going to happen uh, in eukaryotes, usually it can happen with the translocon complex, and which is people have studied with the, in case of yeast it happens with the translocon complex, by the help of another small protein complex called Sec63 complex. So these chaperonins, they are going to move this particular polypeptide chain into the lumen of endoplasmic reticulum by the help of signal sequence. So any of these particular proteins, polypeptide, they are going to carry this N-terminal sequence of amino acids, which will specify that this particular polypeptide chain is destined to be traveled into the endoplasmic reticulum. So this movement of polypeptide chain into the endoplasmic reticulum happens by the help of these chaperonins, which bring it to the translocan, and then translocan is going to open it, and once the translocan gets open, then the polypeptide chain is going to get mobilized inside. Once the first small edge of the polypeptide comes inside, so then a protein called as BIP, which is a ATP binding protein molecule, then it is going to start getting, because it will be attached to the cytosolic portion of the Sec63 complex. You can see in the second figure, so where it is going to be get ATP bounded, then it goes hydrolyzes its ATP into ADP and interacts with the polypeptide chain and pulls it down. See, this is how what the polypeptide chain is going to get completely pulled down, and BIP also acts as a chaperonin. So thereby it protects that linear polypeptide chain, and then later it leaves that linear polypeptide chain with the exchange of ATP for that ADP which is there bound to it. Then leaving that polypeptide chain freely, which can then now be taken care for the protein folding and modifications within the lumen by other protein molecules. So this is post-translational translocation. So now uh, we have studied now how exactly the protein of two origins are going to get synthesized. One, the protein of secretory type, and the protein which is going to function within the cytoplasm, and as well as the one which is actually gets synthesized within the cytoplasm. Now the very important and the final type of protein synthesis is the integral membrane proteins. So integral membrane proteins, these are the proteins which are going to be present completely buried within the membrane. That is, they are going to have their ends distributed at both cytosolic side and at the extracellular side. They are touching both the ends. If you talk about the membrane, means organelles which is carrying the membrane bound organelles, they are also going to have these particular proteins in their membrane. These particular proteins are categorized into four types. In few cases they mention five types also, so where they categorize that fourth type as 5 and 4 and 5, like they further classify this. So mainly there are four of these, one based on the orientation or topology of this particular protein. So topology is a study of this particular topology is nothing but it is the study of structural details of any of these biomolecules. So in this particular case, in membrane proteins, the topology is going to be about, so the how many times these particular membrane proteins they are going to move in and out of the membrane. Then it also discusses like which particular side, that is the which particular terminal, the N and C terminal of the polypeptide chain, where they are going to be present. It also discusses about, concentrates on what is the length of the polypeptide chain which is going to be present freely, in other words, which side, which end, which terminal of the polypeptide chain is the one which is going to have larger size, and the which side is going to be functional. Are both the side of the polypeptide chain are going to be functional, or only one side will be functional, and how do they are going to have the structural organization or arrangement. So based on that, people have actually classified these particular groups. They have made type 1, type 2, type 3, type 4. Type 1, these are the proteins which have normal orientation of NNC terminal. They have exoplasmic N terminal, cytoplasmic C terminal, smaller cytoplasmic terminal, larger N terminal, like example glycophorin, LDL receptor, influenza, HA protein, and then insulin receptor, growth hormone receptor, all this belongs to this category. Type 2 is opposite orientation, opposite to that of type 1. They have larger C terminal domains, and C are going to be exoplasmic side. N terminals are going to be into the cytoplasmic side, and C terminal portion will be more functional there. ACLO glycoproteins, transferins, etc. are the examples there. Third is a one which is having the orientation which is of normal type, but thing is that cytoplasmic C terminal is going to be larger and N terminal exoplasmic side is going to be very very smaller. Okay, so cytochrome P450 is a very good example for this. And then followed by that we have multipurpose membrane proteins. In these multipurpose membrane proteins, here we have categorized into the fourth group itself altogether, but in some cases they made it as 4A, 4B and 5, or sometimes they make it as 4, 5, 6, like that. So fourth, you can see N terminal smaller exoplasmic, C terminal smaller into the cytoplasmic side, and it has multipass membrane protein. Example, G protein coupled receptors or glucose transporters, voltage gated calcium channels, ABC transporters, CFTR proteins, X61, connexins, so many are there. In another kind of protein, it is N terminal outside, C terminal inside, similar kind of thing, only opposite orientation of N and C terminal. There's one more where N and C terminal are going to be both at the same side. That is also there in the membrane proteins. Okay, so these are the few examples or few classification of membrane proteins based on the topology of proteins. Now, how these particular membrane proteins are going to get synthesized? Because as you can see, earlier we discussed about co-translational translocation and post-translational translocation. But in those cases, we have discussed about the movement of polypeptide chain into the lumen of endoplasmic reticulum. Now, this is another process which is going to be called as, which is involved in the movement of polypeptide chain partially into the lumen of endoplasmic reticulum and into the membrane of the endoplasmic reticulum. So, this process is called as co-translational insertion. So it's called as co-translational insertion. Why it's called insertion? Because during the translation process, instead of not just alone translocation will take place, actually it is going to get inserted into the membrane of the endoplasmic reticulum. So that's why it's called as co-translational insertion. So I'll discuss this co-translational insertion I'll continue in the next set of video.